So welcome to episode 31 of Breaking Bio. I'm Morgan Jackson, a graduate student at the University of Guelph in Ontario, Canada. And today we've also got a suite of co-hosts joining us, all from our living rooms because it's bright and early for most of us.
Hi, I'm Rafael Maya, University of Akron, Ohio.
Hi, I'm Tom Housley. I'm a PhD student at the University of Stirling in Scotland.
And our special guest who's joining Tom on the couch today is Stu Auld, who's a professor at the University of Stirling. Well, the research fellow, Stu Auld of the University of Stirling, is here to talk to us all about his research and, I'm sure, various other things as well. So, Stu, what do you do as a research fellow at the University of Stirling?
Well, I'm interested in host-parasite coevolution. And I do a number of different things, so looking at how hosts and parasites co-evolve when there are multiple hosts and multiple parasites in a system. And the research I'm getting on to do at Stirling now is looking at how host sex affects host-parasite coevolution. So I work with an organism that can reproduce both sexually and asexually. And I'm going to be looking at how sexual reproduction causes genetic change in the parasite population. The parasite that infects it is a sterilizing parasite. That's my game. That's my gig.
Cool.
It is intense. Research should be intense, Tom.
So for anybody at home that's maybe not aware, what is Daphnia?
Right, yes. So Daphnia are freshwater crustaceans. They are awesome. So Daphnia magna, the particular species I work on here, is about half a centimetre long, so quarter of an inch, slightly less than quarter of an inch. They have a number of really cool features. So they're transparent, which means you can see their babies developing inside them. And you can see whether those offspring are asexually produced or sexually produced. They look different.
Wow.
When they get infected by parasites, you can see the parasitic infection in the host. So you can go in the wild and collect the animals and see which ones are diseased, which ones aren't, what they're doing. So I think they're beautiful.
Cool.
Yeah,
that's really handy.
See how awesome they are.
Well, you had them on the cover of Ecology and Evolution recently, right?
Yes, our paper, yes. Thank you for the... Go and look for King et al. 2012 Ecology and Evolution. You'll see a...
So, do the parasites that you study have any noticeable effect on the host, like in its behavior or its morphology, or what does it do?
It sterilizes the host. So
okay.
It has a number of key things. So from an evolutionary perspective, it sterilizes the host. So it turns them into zombies. They're genetically dead. It also makes them grow much bigger, causes gigantism, which is interesting. So the parasite causes the host to reallocate resources instead of its own reproduction into producing the parasite and providing more space for the parasite to grow. So,
does it also stop the reproduction of the asexual ones, or just the sexual?
It stops all reproduction.
Oh wow!
With a chemical cue though, so it doesn't physically mess with the host. It just chemically blocks it, because if you treat them with antibiotics, treat the host with antibiotics, they regain the ability to reproduce, which is
cool.
Yeah.
So is the parasite a bacteria then, or what kind of organism is the parasite?
Yeah, it's a gram-positive bacteria.
Okay.
And it infects from the sediment. So Daphnia have this sort of amazing camp action where they waft algal food into their mouth. And they pick up parasite spores as they do this. The parasite is like a bit of Velcro that sticks to the back of their esophagus. and then gets into the host.
How prevalent is this in the wild?
In European, so this is something I did a bit of my postdoc. In Europe, it's really prevalent. So Daphnia magna, it can achieve 80–90% prevalence in populations. It can cripple host populations. In North America, it's much less common. It causes prevalences of sort of 5–10%, because there's another parasite in America that's much more dominant, a fungal parasite called Mechnicalia, and that wins.
So it's not good to be a Daphnia. You've got a lot of things shooting for you, it sounds like, and trying to neuter you.
Yeah, they have a world of parasites trying to get at them. So in that respect, it's not good. It is pretty cool to be able to clone yourself, though. So
that would be useful, especially in academia.
Yeah, it would be. It would be useful. Just have 11 to 20 of you every 3 days. Produce 11 to 20 versions of yourself. Sweet.
Yeah. Pretty soon you would have the infinite monkeys at infinite typewriters, and I could actually write a paper. That would be really useful.
I think I remember seeing on Twitter like a week or two ago that you were preparing some massive, huge experiment, but your Daphnia started all dying or something, like something went kind of astray.
Yeah, I invented numerous new swear words. That was a classic case of setting up a massive experiment, and the thing with my system is when things go wrong, you quite literally pour your work down the drain. You stand there. I had some contamination with the algal food that I feed the Daphnia. So I have these chemostats that grow algae, and some other eukaryote got in there. Something was swimming in there, and yeah, it killed them.
Was that something that you had faced before, like as a student, or was it the first time that something this massive happened? Or
it's the first time that particular things happen to me. It happens to Daphne research. Every now and then it happens. It's quite, well, hopefully, quite rare. It's just a classic experimental system issue.
So it's kind of like you're hazing then.
Yeah, pretty much.
So I was going to ask, you said that they are sexual and asexual. Is it easy to control that for your experiments?
Yeah, so if you keep them in nice conditions, like if they're well fed, they reproduce asexually. If they achieve sort of high population densities, or food is getting low, or they get other signals that winter's coming, so day length, then they produce boys, and then they produce males, and then they have sex and produce these sexual offspring, which look, they're in a different type of egg. The eggs are in a different casing. So you can control it really neatly in the lab, which is sweet.
Wow.
That makes life easier.
It is. And I can do, so one of the things I'm going to be doing soon is I'm going to be hatching sexual eggs and then maintaining the mother and her offspring clonally. And that way I can test a parasite, look at different parasite samples on mum and her babies in the same environment. So obviously one of the big questions in host-parasite coevolution is how does sex mediate coevolution? And the trouble is, it's confounded for time. You're looking at, you can't test things in the past. Well, we can take snapshots of things in the past and examine how parasites have different traits and different genetic composition. That was a very gabbled version of what I'm doing.
So do the parasites affect males and females differently, or is it the same effect?
Well, males tend to, they a big impact on their sperm cells, and so it has pretty much the same effect. But on the population level, because it's such a female-biased population,
Okay.
It's the effects on the females that are most important.
So you kind of just touched on it, but at the basic level, how do you study coevolution?
It's hard. So it's studying the sort of reciprocal effects of hosts on parasite fitness and parasites on host fitness over time. And as I say, the cool thing with Daphnia is that you can maintain them asexually, so you can take snapshots of different time. And something that other researchers have done, Ellen de Carstecker, who is a professor in University of Leuven in Belgium, she has this incredibly cool study where she took sediment cores, and it has the diapause and the resting eggs of the Daphnia and the parasite spores in the mud. So she takes these sediment cores out, she cuts the cores into segments, and she exposes hosts from a particular time, because you can hatch it all out. It's completely, you can hatch them all out again. And she exposed hosts to future and past parasites by looking at shallower or deeper parts of the core. Wow. It's in Nature, and it was one of those really obviously a Nature paper. It's beautifully simple. There's nothing. It's nothing. It's not one of these sort of super clever things. It's just a neat idea with a brilliant design that nails the question.
That's really cool. See, now that's more closer to Jurassic Park shit than anything else.
It's the whole thing. I call it resurrection ecology.
That's all right. Daphnia are cool.
Yeah, they clone themselves. That's way more awesome. I mean, they haven't got any teeth. They couldn't really savage you, but
well, parasites made them giant, so
yeah,
you just need to give them more parasites.
Yeah,
more parasites.
Logical answer.
Really, they continue to grow forever.
Yes, Tom, that's exactly how it works. It could happen to you.
Don't step foot in the fresh water.
The main issue with the freshwater here is that it's seriously cold. Not
So you don't get like your Daphnia from another lab or anything. You go out and just fish them out of the water.
I fish them out of the water. Yeah. In fact, there's a loch just outside the university. By loch, I mean Scottish for lake. Just outside the university, and there have been Daphnia observed there, so I might be able to fish them out like 5 minutes from my office, which would be pretty handy.
That's pretty cool.
Yeah.
So is it all one species, or are you working with different host species, depending on which lake or habitat and stuff like that?
So right now, here I'm working with a single host species, and in the pond, there's just one host and one parasite species. So there's a really tight coevolutionary association. The parasite needs that host. From my postdoctoral work at Georgia Tech, I was looking at different Daphnia species, well, Daphnia and Cereodaphnia, and two different parasite species as well. So there, the parasite has options, and the host has, I say options, but host has different threats of, and you see different dynamics there, which is very interesting.
The expression of sex from, so you said that this is a facultative thing, that they can start reproducing sexually as conditions warrant.
Broadly facultative. I'll come back to that.
Is this related to parasite infection? Like directly?
Well, the jury's out on that. So in the local population I've studied, Daphnia that are more susceptible, Daphnia that have been collected carrying sexual eggs are more susceptible to Pasteurella infection. However, experiments have been done where people have exposed the host to the parasite and then looked at whether, if it's carrying a clutch, whether it has a sexual, it invests in sex as opposed to asexual, found no effect. I want to have a closer look at this because I think it could be to do with when the Daphnia mate. I think if they mate early on in the season, are carrying sperm and then get infected with the parasite, that they shift investment into sex. But this needs to be tested properly.
If they're using this as a response to infection, in a coevolutionary way, trying to generate new genotypes through sexual reproduction, is there any evidence that the parameters of this, like recombination rate or such, are different, like comparatively across species that are, infected by these parasites or not?
I don't know. So people have looked at, so there are some Daphnia species or some populations of species. So there's a population of Daphnia pulex that's wholly asexual in northern Canada. And researchers have looked at what parasitism, how often they're infected there, and haven't really found much interesting there. The thing about the Daphnia here is they have sex. They tend to have sex once a year. So towards the end of the season, they reproduce sexually to survive the winter. So they do recombine each year. It's just a matter of when they do it.
I'm pretty sure that's the hardest question anyone's been asked on this podcast.
No doubt. It also leads that I feel like there's all sorts of jokes about comparing the sex lives of Canadians versus Brits based on their Daphnia species. But we'll leave that for another podcast, I suppose.
Yeah, we can ask you back on in the future.
Yeah.
So I guess kind of shifting gears a little bit, but I was curious. So you've got an appointment as a research associate, right? I get it right?
It's this classic British system thing. So I'm a research fellow.
Okay.
So it's kind of like a postdoc, but I'm my own boss. So it's kind of halfway between being a postdoc and an assistant professor, really.
Okay.
It falls in that middle ground.
And you started that this year, right?
Yes, in January. Yeah.
So I was kind of curious, and I wonder if you could speak about a little bit about how that transition went. I mean, so you were, recently a student or a researcher working with an advisor in the lab, and now you're kind of setting up your own stuff. And so I was wondering how that went. What did you find of it?
Well, apart from everything dying, I've got that sorted now. It sounds like there's a big jump there, but it's much smoother than it first appears. So in my postdoc at Georgia Tech, I work with a great boss called Megan Duffy, and she gave me quite a lot of independence in the lab. So I was working with the same parasite I did my PhD with. So she let me have playtime, essentially. It was like, you do what you want. and it was really good. And then moving here, although I'm my own boss and I'm setting up a Daphnia lab, I'm working closely with other individuals in the department, mainly Matt Tinsley, who does work on immune responses and senescence in Drosophila. And he's my research mentor.
Oh.
So I still have a lot of support. It's just it's more of a graduated thing.
Uh-huh. And are you taking on students or stuff like that, or are you working on your own?
I have undergrad starting, but I'm working on my own for now and writing grants and trying to win money to support students. And
That's
then
really cool.
I hope to get.
And your postdoc was in Georgia Tech,
Yes,
which is here in the U.S., right?
it is.
And what was your PhD also here in the U.S. or?
No, my PhD was closer to here, actually. I did my PhD in the University of Edinburgh, Scotland, with Tom Little, who is a Daphnia guy. So I've been working with different species of Daphnia throughout.
Oh, cool.
Just to mix things up a little bit.
Yeah. My biggest jump was working with American Daphnia, which, yeah.
That's crazy.
It's crazy. System.
But do you have to get to know all the different parasites then, or do they work in similar ways?
They're quite different. Well, so the North American, there are two main North American parasites. So there's Pasteria, but that's not as big a player. And the important one is Mechnicauia bicuspidata, bit of a mouthful. That's a yeast parasite fungus. That has, so it doesn't sterilize its host, but it kills them earlier. So it has its virulence is expressed in different ways. If you wanted to find out more, there's Auld et al. 2012 in one, which compares sort of infection genetics and virulence between Mechnicauia and Pasteria. Yeah. Just shameless plug there.
That's fine. That's what we use this podcast for all the time. It's one continuous shameless plug, essentially. So it sounds like you've got a pretty nice step up. Like, you hear a lot of labs where they go from postdoc, of varying degrees of individuality, sometimes not very much, being thrown into a faculty position where suddenly you are right in the deep end of the pool, and you got to swim or drown.
Glory.
To get back to the Daphnia example, I guess. So it sounds like the British Research Fellow program or system, I suppose, is kind of like that extra intermediate step that I imagine is going to be pretty helpful for that career ladder.
It is, and it is. So I like, the big issue with it is, so my appointment is for 2 years, and then in that time I can either win another research fellowship or try and convince the university to make me make me a lecturer, just like assistant professor. So it does just delay the permanent job part.
Mm-hmm.
So I think it's a good system, and I like the graded levels of independence, and that's great. And I'm not required to do any teaching. I will do because I enjoy it, but I have control over how much I do. But there's still, as we all have, that permanent job.
Yeah.
Still waiting for that.
So you still have death or glory right now?
Right now. My whole life is death or glory. I'm enough on the edge.
So far, mostly glorious.
Except for that colossal amount of death.
It's all.
It happens.
So, anybody else have anything to speak about for Stu? Stu, do you have anything you'd especially like to get out to the world, get off your back? I don't know, break any or burn any bridges live on air?
Burn any bridges. A list of people in the field I don't like.
That works.
Excellent.
This is where you get to watch me just ransack my career on screen.
That's what we're here for.
No, glory. No, I had a paper accepted yesterday, which I'm excited about, that
Nice.
I will tweet vigorously on in times to come, on Parasite Evolution in the wild, which will be in Oikos. So yeah, watch this space for that.
Can you tell us about it, or is it
Yeah.
a little bit under wraps?
Well, Oikos aren't going to sue me. So I go out in the field and collect parasite samples from across the season, collect Pasteria samples. And the good thing about Pasteria is you could keep it in the freezer. It doesn't affect it. So I took parasite samples from across the season, put them in the freezer, and then exposed them to a standard set of host genotypes. So I took Daphnia and exposed these parasites from different times. and found that the parasites become more infectious and they have higher within-host growth over time. But, interestingly, parasites collected at the end of the season weren't as virulent. So they infected more hosts, they rocked out when they were inside the host, but they didn't cause the host to die any earlier. Which is quite odd, because evolutionary theory predicts that parasites that grow fastest kill the host fastest. And we didn't find that. So we're still trying to work out exactly why that's going on. But yeah, so it's quite a simple study, but what we did is showed wild parasite evolution. And a lot of what you see on parasite evolution is on evolution of antibiotic resistance or other such things in the lab, whereas we've got it happening in a co-evolving system.
How are the parasites? How are they transmitted?
So they're horizontally transmitted. I was saying earlier that I think before you managed to tune in, the Daphnia sort of waft algae into their mouth when they feed, and they pick up these transmission spores of the parasite at the same time. And it infects via the gut. And whether or not the host gets infected depends on whether the parasite can stick to the esophageal wall. And then once inside the host, it proliferates and then is released when the host dies.
Yeah, sorry, I was just thinking that high rates of horizontal transmission could explain why your virulence or your growth rate doesn't necessarily translate to virulence.
Yeah, so in the field though, at the end of the season, the incidence of infection is much lower. The prevalence is much lower, and that is because, as well as the parasite population evolving, the host population is also evolving. So in the lab, we've uncovered parasite evolution, get this increase in infectivity within host growth. In the field, you've got parasite evolution and host evolution as well. Hosts at the end of the season are more resistant than hosts at the beginning of the season. So there are fewer infections, but the parasites that got into those few infections are better able at getting in once you control for...
Well, if people want to learn more about your research and follow along with your Daphnia, where can they find you online?
So, best place to look for me is at www.stuartauld at wordpress.com. And I have just a simple website. I'm on Twitter. I'm at Stu Auld on Twitter, where you can hear me express my many opinions about the world. Yeah, that's me.
Awesome. So I guess we'll wrap up now that you're getting backened off. Thanks a lot for joining us, Stu, for episode 31 of Breaking Bio. It was a great time. And I'm sure you'll have to come back later when your Daphnia are all alive and you get some new research. So we'll talk some more.
I'd love to talk to you guys again. Yeah.
Awesome.
Thanks.
So, thanks again, and thanks for joining us for episode 31 of Breaking Bio. You can find us online at breakingbio.com or on Twitter at breakingbio. And we'll see you everybody again soon. Bye. See ya.
See ya.